Okay, so now we've walked through how to edit and generate reactions and how to edit and generate compounds. So let's take a look at what we can do with proteins. So we're going to go up here, protein, and let's just pull up aspartate kinase. And here we are, aspartate kinase, lyse C in this case. We're pointing to the reaction that we were looking at earlier. Now, as we mentioned before, Pathway Tools is set up to have most of the functional curation for gene products in the protein pages. Our expectation is that you're going to put the what this thing does into the protein page rather than the gene page. So we have a lot of tools to help you do that. You probably won't be making a lot of new protein monomers over time in your database because we've generated them off of the genes that are already there. But if you need to, you can. You might, for example, need to make a protein monomer that's not connected to a gene to represent an enzyme whose activity has been assayed and even maybe has been purified in the past, but for which you don't have sequence information. And this actually does happen a fair amount of the time. So let's go up here onto the name and right-click to see what our options are in the editor. Okay, so we have the protein editor, which is the interface we'll be using to edit the protein. We have the protein subunit structure editor, which is for protein complexes, which we'll address in a little bit. And then other options that you basically don't want to touch again, like the frame editor. You might want to use the synonym editor that lets you change the name of the protein, other things, without opening the main protein editor. And there's a add feature thing, but you can actually add features within the protein editor. So we're basically going to want to stick with the editor. Now, we're looking at the inside of the protein frame. Let's see what we can change here. Okay, we can edit the enzyme name up here at the top. We can add an evidence code for a non-enzymatic function. We actually have a different evidence code slot for enzymatic reactions, for small molecule reactions. And the evidence code thing here, all you need to do is click on evidence code and it will give you this menu of options. And our full evidence code is explained in our documentation and on the website. But say, for example, that we had a non-enzymatic function that had been determined experimentally with purified protein. For example, if aspartate kinase were shown to also be a chaperone that promoted protein folding, then we could put a citation in here. And the basic point of evidence codes is to say why we believe that something does what we say it does, and to let you associate that citation in a structured fashion. Now, citations you can add in the space available here. You can add them as Pubmed IDs, and that's the most straightforward way. If the thing you're citing does not have a Pubmed ID, then you can just type something in. I will type random junk. And when I click away, it's going to tell me, well, that's not the correct format for a Pubmed ID, and it's not a frame in your database already, so do you want to make a frame? And we say, sure. And we say title is random junk paper, and author name is author A, and source Journal of Generic Research. And you can put in all these things like that, and you can say OK. And so now we have a mnemonic reference there. We could also just as easily have gone in and put in a Pubmed ID. So let's try one of those. And when you put in a Pubmed ID and click away, it's not going to bother you about it because it doesn't really, it looks at it, the software looks at it and says, this looks like a Pubmed ID, great. Now, when I hit OK and come out of this, and I'll do that right now, then it'll have an evidence code up here, and you can see that it's given me this little pop-up tooltip telling me what evidence I say is there for this non-enzymatic function, and it includes an assay of purified protein, and a mnemonic for the author, and I helpfully called the author author, so it's showing up here as saying author. Now, I'll get into this a little bit more in a moment, because we're going to move down from here to gene classes, go terms, and the summary. So let's talk about gene classes and go terms for a little bit. Gene classes lets you choose from components of the multi-fun ontology, which is a protein functional ontology. And you may or may not be entirely familiar with that, but it has the kind of things you'd expect to see there. And for example, this could be plasmid related. I kind of sincerely doubt that it is, but it could be. Now, also over here you have Go terms, gene ontology terms, which you may be familiar with. If you're not, the Go Consortium has a full explanation of Go terms on their website. And we help you out here because you, like almost any curator or researcher who is not a full-time member of the Go Consortium, may not know, the Go hierarchy off the top of your head. It's large, complex. So if I click here on Go terms, I get a browsable hierarchy, so I could just click through and select stuff. But I also get a search window here. And so, for example, I could type in aspartate kinase, and I could click search, and you have to click search. Hitting enter here, as you may be used to doing, won't work. And what the software is doing now for you, and it's done, is searching through the entire Go hierarchy for appropriate terms. And we see, aha, aspartate kinase activity. And when I click on that, it's highlighted up here, but I haven't yet chosen it because I may want to browse around within the hierarchy there and see if there's something more appropriate. For now, I think aspartate kinase is a perfect hit, this Go term here, 4072, so I'll select it. And now it appears down here under selections. And you may notice this window here is actually showing you the definition of each Go term as I mouse over it. But so we've selected aspartate kinase activity. Great. We'll leave it at that for now, though usually you'll be attempting to go for a number of Go terms on each protein that you're editing. I'll hit OK. Now, following the Go term, we have the ability to put in an evidence code, which is a critical part of assigning Go terms. And the evidence code, typically is going to be the same evidence code you used for the determination of the activity, whether it was enzymatic or not. And so purified protein again, and we can put in the same Pubmed Id citation that we had before. So, and then if I again hit OK, I see that, okay, the Go term now appears on the protein page, which is very exciting. And there's this little number 2 there. And that says, well, there's an unimported citation there. So how do we solve that problem? We go up to File, Import, Citations from PubMed. And now we click that and then watch. And you can see the process is going on down here in the lower part of the screen. Oh, and there we go. So, since this is the first time I've done this, wow, 79 publication frames and 15 references that couldn't be found. We could look through those right now. This is actually a good part of database cleanup. This will tell you, hey, these references may or may not work, and we're not going to worry about that now, but what we do is we hit OK. And then you see it still hasn't shown, and that's because we haven't, say, clicked away from the page and clicked back on it to let it re-render. Or just clicked on itself, and aha! Now we have this mnemonic representation. In this case, CASPI 07. I actually cited us. It's the most recent MetaPsych paper. And so we have this mnemonic representation with the name and the year, and you can have other representations as well. You could have just a number, you can have other formats of representing the name. These are all set under Tools, Preferences, and they're just like setting the colors for the different things, the different objects in the display. It's just preference setting stuff. It doesn't have to do with editing. So now we've put in that name, we've put in stuff up here. We might want to start saving changes. In fact, I recommend saving frequently just in case something funny happens with your computer. I'm a habitual saver. So to save, we have it easily up here. We just click Save DB, and it will tell us that it's saving. And that's it walking through the database and saving our current changes to it. And now we're saved. Okay. And you can save very frequently. You can't save while you're in one of the editors. That's important to remember. So I recommend, just on the off chance that something bad happens, not doing a whole ton of unsaved work all at one time in the editor. I'm happy to click out of the editor, save, open the editor again, keep working.